Hello all. In this presentation we are going to see about entomology, how to identify entomology spotters. So we all know there are two kingdoms, animal and plant kingdom. We belong to the vertebrate, in that we belong to the mammals as a class. So like that phylum classification, there is arthropods. Entomology is study of arthropods or insects. In that again class Crustacea, Arachnids and insect are there. Medical entomology is study of arthropods of medical importance. So these are all the list of arthropods of medical importance. So these arthropods are the vectors of the disease. Mark my words, vectors for the disease, not the causative agent. That is, these are all the species which just transmit the diseases, out of which housefly transmits the disease by mechanical route, and the other vectors transmit biologically. That is, the agent undergoes some either multiplication or change in form within their bodies. Now these are all the few background knowledge which you should know before going into the identification of the entomology spotters. Here it is broadly divided into Insecta, Arachnida and Crustacea. Insecta has head, thorax and abdomen. Arachnida and Crustacea has both thorax, that is head and thorax get combined. So legs, here we have 3 pairs, 4 pairs and 5 pairs respectively. Antenna is 1 pair, 0 and 2 in Crustacea. Wings are 1 or 2 pair of wings. Arachnida there are no wings, and Crustacea there are no wings. When come to the habitat, Crustacea lives in water, the other Insecta and Arachnida all live in land. So these are all the group of insects which are all of medical importance. This is cyclops, ticks, mites under Arachnida, mosquitoes, flies, lice, fleas, redweed bugs among Insectae. So here this is mosquitoes, we all know, and majority of the presentation we will be discussing about mosquitoes. So these are all flies. The notable one are housefly, blackfly and CC fly. Housefly is a mechanical vector for many fecal-orally transmitted diseases. Blackfly transmits onchocerciasis, CC fly sleeping sickness. This is housefly. And then we have louse or the head louse. Then we have rat flea. This is the classical image of the rat flea, from which we can identify the rat flea spotters easily. And this is the redweed bug. Then we have ticks and mites. So when there is a head separately present and almost conical, then you can say it as a hard tick. When this is of exactly a cuboidal shape or a smoothened rectangle shape, then it is a soft tick. Then we have itch mite and cyclops. Cyclops, this is we have 5 pair of legs, 2 pair of antennas, and there will not be any wings present. So they live in water. Cyclops were the vector for guinea worm disease. So here is the list of arthropod, that is mosquito, housefly, sandfly, CC fly. Here is the list of arthropods and the list of diseases. So mosquito transmits malaria. Malaria specifically Anopheles transmits malaria. Viral encephalitis, Japanese encephalitis, filaria and all is transmitted by Culex mosquitoes. Yellow fever, dengue, chikungunya and all are transmitted by Aedes mosquito. Then we have all this housefly, which is a vector for transmission of many fecal-oral infections. We have sandfly, which transmits cholera, oriental sores, and CC fly transmits sleeping sickness. Louse transmits pert, that is pediculosis, epidemic typhus, relapsing fever, and trench fever. Rat flea transmits plague, bubonic plague, endemic typhus, Hymenopsis diminuta, chickerosis. Black fly transmits onchocerciasis, redweed bug, Chagas disease. Soft tick, Q fever, relapsing fever. Hard tick, there is a huge list of diseases. Scrub typhus and rickettsial pox are transmitted by trombiculate mite. Scabies is transmitted by itch mite. Guinea worm disease and fish tapeworm by cyclops. Enteric pathogens by cockroach. So when we are identifying, we have to write 3 important points. We have to write the basic morphology of those spotters. Then second is the diseases which is transmitted. Third is control measures. How are we going to control it? So these 3 points we need to write under all the spotters. So first and or the easiest spotter is the egg. We have to differentiate between Anopheles, Culex and Aedes. So when we mention Culex, C is the mnemonic here. So we have to say Culex will lay egg in clusters, and individually the eggs will be cigar shaped. So when the egg is clustered, you can easily identify it as Culex egg. When the egg is singly, it may be either Anopheles or Aedes. When it is a spindle shaped, it is the Anopheles. When it is oval shaped, it is Aedes. It has lateral forts, that is, air will be filled in it so that it will float. The Anopheles basically is a surface feeder, so it has these lateral floats, and once it becomes larva also, it has no siphon tube. It is floated parallel to the surface of the water, and it is a surface feeder. This Culex and Aedes are the underground feeders. So now we are moving to the larvae. How to identify this larvae is by this siphon tubes. Siphon tubes are otherwise the respiratory trumpets through which it respires. So when it is very apparently present, conspicuously present, then you can say it is a Culex larvae. When it is present, not as long and slender as Culex, then you can call it as Aedes. When it is entirely absent, then you can call it as Anopheles. Another finding to confirm Anopheles is this presence of this palmate hairs. While noting this siphon tube, we should not get confused with this anal gills and caudate hairs for siphon tubes. Sometimes these will get clubbed, and in slide it may appear like a siphon tube. So we should not get confused when we are identifying a larvae. Then pupa. In real life scenario, both this larvae and pupae or eggs and all species can be easily identified based on the water source, the type of the water source which is present. Based on that, we can easily identify the species. But here, out of these stages, that is egg, larva, pupa and adult, pupa is the difficult stage to identify. So when there is no siphon tube, again it is Anopheles. When there is a apparently long and slender, then it is a Culex. When it is ill developed, then that is a Aedes. So this is not the typical microscopic image. Now we are moving to the adult mosquito. So when we are identifying the adult mosquito, when you identify the spotted wings, then you can directly say it as Anopheles. When you are seeing these spotted wings, then you can say it as Anopheles. When this is not present, you have to look at the body. When there is alternate bands of black and yellow bands are present, then you can call it as Aedes mosquito. When both the spotted wings and these bands are absent, then you can call it as Culex mosquito. How to identify a male is by this means of the bushy antenna. So before going to the antenna, you have to understand the basic structure of the head. From head, there will be a central proboscis through which it sucks the blood, and palpi, immediate lateral palpi, further lateral antenna. So this is proboscis, this is central proboscis, the immediate lateral palpi and the antenna. The antenna will be bushy. This is corresponding to the moustache in males, bushy in case of males. It is hairy, but it's not bushy in females. We can identify in the next upcoming slides. This is for the female species identification again. So when we identify this Anopheles, spotted wings, we can directly say it is Anopheles. We have to look at the wings. So wings will be very light in Aedes. It will be very heavy in Culex. And because of these heavy wings, these mosquitoes are called as nuisance mosquitoes. Because it makes noise when it flies near to the ears, that people misinterpret it as the mosquito sing some songs. Anopheles palpi is very short in case of female. This is the palpi here. This will be very short in females. The only exception is the Anopheles, which has long palpi. We can easily identify when the palpi is short and the antenna is not bushy, then it is a female. Spotted wings and palpi is big, then that is a female. But in the corresponding male also will have a boat shaped or a club shaped palpi will be present. So with the wings and the body, we can identify. Body is same as Anopheles will have that band, but here this slide is more dark, so we could not differentiate. But by through wings we can easily identify. Then we are moving into the mouth parts. As I told earlier, all the females will have very short palpi and a less bushy antenna. Here it is a bushy antenna and it is a lengthy palpi as much as your proboscis. Then in Anopheles is the only exception where the palpi is also big when compared to the proboscis. This is the hair is bushy. The hair is bushy. The hair in the antenna is not bushy, but here in the hair in the antenna is bushy, and here it is club shaped. So when the palpi is club shaped and a bushy antenna, then you can call it as an Anopheles male. When the bushy antenna is not bushy and the palpi is big, it is the usual size will be till here, but it is till here means then you can call it as an Anopheles female. So that's how we can identify the Anopheles mouth part. Then there are some other insects other than mosquitoes. So this is the Cyclops, belongs to the class Insectae, and sandfly, which belongs to the Insectae. So this looks similar to the mosquito, but when we look out of the microscope, the size will be almost half of the mosquito, and also it is a hairy animal, and comparatively the legs are very bigger when compared to this body. So then you can suspect of sandfly, and you can note it as sandfly. This is the head louse. These are all classical pictures. The morphologies are very clear. Then ticks, soft ticks and hard ticks will be confusing. It has 4 pair of legs. When the head is out, it is a hard tick, and the shape is also little conical. And here the shape will be rectangular. After a blood meal, this will be very much apparent. Then we are moving to the rat flea. This is the classical image given in textbooks. So it looks similar to this. We can easily identify if we remember this rat flea pictorially. So hopefully this presentation was useful to you. If you have any suggestions and doubts, you can use the comment section. If you haven't subscribed, please subscribe and click on the bell icon. Thank you.